Okay, so today we are going to talk about a different way to model and simulate biological systems. And mostly it's a new way to simulate, because the part of the defining the model is very similar to what we did with the differential equation systems. But here we are going to consider that instead of assuming that all the processes in the system occur at a well-defined rate given by the differential equations. Can you hear me correctly?
Yes.
Yes, yes.
Because my image has frozen. Yeah. Okay. So in stochastic simulations, we are going to assume that although the average rate of a process is defined by an expression, at each instant, at each moment, the rate will be affected by random events, by random chance. So that's the difference, the main difference from the systems of differential equations. So what is the main difference is that what we were using, systems of differential equations, are deterministic systems. And by deterministic, what I mean is if we start a simulation at a given initial condition, the trajectory of the system is going to be exactly the same every time. While in a stochastic model, even if you start the system exactly with the same values for every dependent variable, the trajectory of the system can eventually be different every time we do a simulation. That's because at each time step in the simulation, we are going to use some kind of random number generator to simulate what happens in reality in these systems. Let me check if this is strange. Sorry. Just sorry. So the deterministic system defined by differential equations works because it assumes that the rate expressions are valid if we are considering a large number of molecules in a well-mixed environment. So if we say that a given molecule in our system has a concentration of 100 millimolar, we assume that in every sub-volume of our physical space, that concentration is valid. So there is no spatial heterogeneity, and we have a big number of molecules. And in that way, statistical mechanics assure us that the rates that we are computing with the differential equations are a very good approximation of what is happening in the real system. But if we want to model a system with a small number of molecules, then the stochasticity of the chemical kinetics becomes important. Why? Because all these chemical reactions or binding and binding processes that we simulate in biological systems, they happen because molecules collide with each other. And that happens because molecules have some kinetic energy, so they are moving in space randomly with a random direction, and the velocity of that movement is a function of the temperature of the system. And so a reaction occurs if a given, with a rate that is proportional to the number of collisions, of effective collisions that happen in a given unit of time. And if we are considering a large number of molecules, the differential equations provide us with a good approximation of the overall rate. But if we are working with a smaller number of molecules, we have to compute not the rate, but the probability that a collision will occur at a given time interval. So that's where chance comes in. Although every time we do a stochastic simulation, we get a different trajectory of the system. For many systems, especially if we make a stochastic simulation for systems with a larger number of molecules, the average of all the stochastic trajectories will correspond, will match with the solution of the system of differential equations. So if that is true, and that happens many times, why it is relevant to make stochastic simulations? That is, if we make the average of a lot of stochastic simulations, we get solution we get from a system of differential equations. What's the utility of doing stochastic simulations? Here is an example of a simple system with it's basically a decay of a molecule that is being degraded with a simple mass action kinetics. It starts at a given initial value, and so it decays. In the beginning, the decay is more as a higher rate because the decay rate depends on the amount of the molecule that is being degraded. And the curve in black is the result of the solution of the differential equation. So it is a very well-defined time course. And with these three colors, we have the stochastic simulations of the same system. And you see that, for example, in the green simulation, it happens that even in the beginning, you have fluctuations. So there is the rate of degradation is not always constant. But if we are only seeing three simulations, but if we made hundreds of simulations, eventually the average of these stochastic simulations would start to converge to the solution of the continuous differential equation system. The main difference is that in the differential equation system, the decaying variable only reaches zero in either at infinity time if this is converging very slowly or after a very long time. While in the stochastic simulations, just by random chance, some of these simulations will reach zero. And once the variable reaches the value zero, it will not be able to decay anymore. Okay, so that is something that you never see in systems of differential equations, but in stochastic simulations, you may observe events that we can call extinction events or catastrophic events, where by random chance, the values of the variables reach either a minimum or a zero or explode to a very high value. And that will correspond in the biological system to, for example, to a death event, and that you are not able to see in the systems of differential equations. And so we can compute a lot of simulations and measure, for example, how frequently you get these stochastic extinction events, instead of being interested in the trajectory that you can, the average trajectory you can calculate with the continuous differential equation system. Here is another example that this corresponds to what we saw when we talked about circuits that present oscillations in biology, and we talked that systems with that converge to a stable steady state by damped oscillations, they can present sustained oscillations if there is noise. So in this case, that's what it is. In this upper graph, we have a system that the red line is the solution of the differential equations. So it is a very smooth damped oscillation, but it is. So it goes up, goes above the fixed point, goes below the fixed point, and then finally stabilizes at the fixed point. But in the stochastic simulation with noise, so with the random stochasticity is not a good expression. So with stochasticity, you observe these periodic oscillations. They are not perfect oscillations. They have a noisy shape, but the system will not converge to a very stable fixed point. And in practice, in stochastic simulations, you almost never have perfect fixed points because maybe the system is fluctuating around the fixed point of the system, but it is always changing.
Could I ask something?
Yes, sure.
But this noise, it's always there, right? I mean, there's always noise, or is that not true? Like in practice, there's always noise.
So
right?
in practice, there is always noise, because we are going to talk about that. I'm not sure if it is in the next slide or not, but
Okay.
but there are different sources of noise, and they are always present. The thing is that if you are looking at a system that has a large number of molecules, at least one of the sources of the noise is cancelled out. And so
because of it's just being an average of everything.
yes, if you have large numbers, then the rates of the processes converge to the solution of the differential equations. And in practice, in reality, this system is very well approximated by the continuous model.
Okay, thank you.
But even in that system, if we look microscopically, there is noise there, and there is stochasticity in the rates. But as you have large numbers, those fluctuations, noisy fluctuations, cancel out each other. So this is another example of things that we can see that are different in stochastic simulations compared with the systems of differential equations. And it is another example. We saw systems of differential equations that had multi-stability. So they have more than one fixed point, for example. And when we performed simulations in those bistable systems, we started at different initial conditions, and depending on the initial conditions, the system converged to a different fixed point. But for the same initial conditions, the fixed point that was reached was the same always. Because in deterministic systems, you have you have the attractors, and each attractor has a basin of attraction. And if you start at the same position in the system, the same state, you will end up in the same fixed point. But in stochastic systems, you can start in the same initial condition, but due to these random fluctuations, some of the variables may cross over the basin of attraction, and some simulations will converge to one fixed point, while another proportion of the simulations will converge to a different fixed point. And that is also a possibility that is also observed in some biological systems. So noise is a way to. So we didn't. So when we talked about bistable systems, we thought that that was a way of biological systems to make a decision and having some kind of memory. So some signal pushed the system to a different steady state, and the signal was gone, but the system kept in that different steady state. But with noise, if we are working with a system with a low number of molecules with that bistability property, what we'll have is a stochastic choice of the fixed point. And depending on the size of the basins of attraction, one of the fixed points will be observed more times than the other. And that can be used by biological systems for their benefit, and we are going to discuss that example in a minute. Yeah, this is another situation that before we were talking about the differences that, for example, here some of the simulations went to extinction, sorry. Here we observed oscillations, here we observed a consequence of bistability, but even in the same simulation, a system that has multi-stability may show something like this. So this is simulates the protein levels of a given protein that is being expressed and synthesized and degraded, and it has apparently three more or less identifiable possible steady states, one with a high value of protein copies, one intermediate and one low. And given due to noisy events, you can migrate from each of these different states. And if you make a histogram of all the values of protein levels across time, you may observe something like this. So this is one that does not correspond to this picture because it doesn't have this middle state. Actually, this is a different parameter, right? You can observe this multimodality that corresponds to different quasi-fixed points. So they are not real fixed points because they are always changing, but you see that when the system reaches that region, it tends to stay there for a while, but then another stochastic event brings it out of that state, and so on. And so in stochastic simulations, you are not only interested in the shape of one trajectory, but in accumulating either a long trajectory and study its distribution of states possible through time, or conducting multiple parallel simulations and studying their statistical properties. So when what is the rule that we follow to choose if we should perform some kind of stochastic simulations instead of going with the continuous system of differential equations. So, because in theory, all biological systems are exposed to noise and constantly. That is a that is um, that is accepted, that is true. But we have fundamentally two sources of noise. We have extrinsic sources of noise and intrinsic sources of noise. So the extrinsic sources of noise are stochasticity that arises between different cells. So we are assuming that all biological processes, of course, there are biological processes that occur between, that involve the communication between different cells. But if we are thinking for now in processes that occur within cells, there are some processes that are different from cell to cell. But it doesn't mean that the system has a low number of molecules, for example. But as cells divide, they can be at different stages of their cell cycle. The environment that each cell is sensing may be different. And after cell division, the two daughter cells may have a different number of organelles each, or for proteins with a smaller copy number. For example, a protein has an average copy number of 100 copies per cell. And that cell divides and gives origin to two daughter cells. It is, of course, on average, each daughter cell gets 50 copies of that protein. But as it is the spatial division of the cell into two daughter cells is not completely controlled. So the division of the genetic material is highly controlled, but the other components of the cytosol are not so well controlled, the division between the two daughter cells. So it may happen that one daughter cell gets 25 proteins and the other 75 of that particular protein. And so those two cells will start in different states. So their trajectories of the processes that are dependent on that protein will be different from cell to cell after each cell division. And that is a source of noise. So when we look at measurements in biological cells, we get a variability, a high variability, a random variability from cell to cell that may be due to these extrinsic factors, either variations in the environment, in the cell cycle phase, and in variations in some cell components that are due to asymmetries in cell division. So if you are only interested in studying how the biological systems adapt, or how do they deal with these sources of extrinsic noise, you can still use continuous systems. So you can still use ordinary differential equations. That what this acronym means, ordinary differential equation systems. You can still use them because most of the times you can model these extrinsic noise, extrinsic sources of noise by performing multiple simulations of your system of differential equations with different, for example, with different initial conditions, with different parameter values, or with different independent variables values. So that you insert the noise in the value of the parameters, for example. So you make a collection or a large number of simulations where you assume that the one parameter of the model has a normal distribution with a given variance, and a given average value, a given average mean, and then you perform hundreds of simulations varying those parameters according to that normal distribution. And that is a way that you can study what is the effect of the extrinsic noise and have and measure or quantify what different behaviors can arise from cell to cell due to that extrinsic noise. But all the systems are affected by intrinsic noise sources that are affect even within a single cell. If you look at a cell during a period of 10 seconds and after those 10 seconds, you observe the cell for another set of 10 seconds, the rates of even if the cell is approximately in a steady state, the rates of the different processes, especially the ones that have a smaller number, involve a smaller number of molecules, in the first period of 10 seconds may be quite different of the rates of those processes in the next period of 10 seconds. So that variation within the same cell is due mainly to a low number of molecules. So those collisions that give rise to the chemical events, to the reactions, become, so the stochastic factor of those collisions becomes more dominant. So you cannot predict accurately how many events are going to happen in the next second, for example, because the probability that the molecules collide or not is not very precise. It gets precise if you have a larger number of molecules. And so that the effect of this intrinsic noise is only, we are only able to study the impact of this intrinsic noise by performing stochastic simulations. And why are we interested in studying the impact of noise? So, as we said, noise is present in all systems with different sources, but it's present in all systems. And generally, we study noise with two main perspectives. So many times noise is seen as something that is not desirable for the biological system, because we have observed that biological systems tend to maintain some of their internal variables constant. It's a property that is called homeostasis. So it's a property of living things, living organisms, that they usually tend have mechanisms to maintain their internal state. So we are not always changing. And so biological systems must be designed or have evolved to be robust to these noise sources. So even that the noise is there, it is present, the way the system is organized, the way the system works, it is robust, so it performs the same function, but independently of the effect of noise. So that's one of the approaches. So we want to make stochastic simulations to check if the system with that properties, with those regulatory interactions, with those reactions going on, is it robust or not? Because if it is not robust, then that system maybe is not an accurate model of the biological system. But sometimes it's not the majority of times, but in some situations, biological systems have evolved to take advantage of noise. So they use noise to increase their survival probabilities. And one classical example of this exploitation of noise is in bacteria that have antibiotic resistance genes. So we live with a lot of bacteria in our gut and in our skins, for example. So there are more bacterial cells in a human organism than human cells. mostly in the gut, and live with them happily. But sometimes some of these bacteria are bad for us, so they are pathogenic. And we have developed drugs called antibiotics that allow us to fight those pathogenic bacteria. And since we developed antibiotics, some bacterial species have evolved genes that allow them to survive even in the presence of antibiotics. So those are resistance genes. So those are genes that, for example, they metabolize the molecule of the antibiotic. So they degrade the antibiotic and it no longer is able to kill bacteria. Or there are molecules that pump out the antibiotic out of the cell, reducing the intracellular concentration of the antibiotic and therefore minimizing the effect of the antibiotic. But those genes that allow bacteria to survive antibiotics have normally a metabolic cost. So if you have two bacteria, one is expressing the genes that confer resistance to the antibiotic, and the other one does not express those genes, the one that expresses spends more energy synthesizing the proteins of the resistance codified by the resistant genes. The other strain is not spending energy, metabolic energy, producing those genes, those proteins, sorry, so it will grow faster. So it will have a higher yield in biomass production because it doesn't have to be spending energy and building blocks like amino acids to synthesize the resistance proteins. So it will grow faster. So it will, after some time, there will be a much larger number of the bacteria that do not produce the proteins that confer resistance. That is true if there is no antibiotic in the media. But if you have antibiotic present in the media, that will kill the bacteria that do not synthesize resistance proteins, and the bacteria that synthesize resistance protein, although they grow slower, but they are the only ones that grow. So they will dominate the population after some time. If the regulation of the expression of the antibiotic resistant genes is the simple regulation, the bacteria either synthesize them or not. And that is, so that means that the bacteria that synthesize proteins that confer antibiotic resistance will only be able to survive in environments with antibiotics, because only then they can win the competition with other bacteria, the growth competition with other bacteria. But antibiotics are not present all the time in the environment. So some bacteria have developed a way to regulate the antibiotic resistance genes that is a system with bistability. So the regulatory circuit that regulates the synthesis of the antibiotic resistance genes, proteins, has two steady states, one high steady state and one low steady state, like the positive auto-regulation systems that we have studied. So it's a situation like this one. So there is a fixed point with a high level of the protein and a fixed point with a low level of protein. In this case, it would be helpful that this lower level would be much lower, so that metabolic cost would be very low. And after each cell division, there is this stochasticity stochastic choice, and that particular cell will either converge to the low steady state, so it will not produce the antibiotic resistant genes, or by random fluctuations, it will end up converging to the high steady state, and it will produce the antibiotic resistance genes. And normally, if most of the times the bacteria is living in an environment that doesn't have antibiotics present, then that system will be tuned up, in a way that most of the times the system will converge to the lower production of antibiotic resistance, and only a small fraction of cells will be producing antibiotic resistance proteins. So the thing is that once the antibiotic appears in the environment, some of the cells that are dividing will always give origin to some cells, a small number, that produces antibiotic resistance genes. So if the bacteria is in the presence of antibiotics, at least some of the new cells that are produced after cell division will be able to survive in that environment, and the other ones will be killed by the antibiotic, will not survive the antibiotic, but a fraction of the population will give rise to survival of the species. When the antibiotics goes away, so it is not applied anymore, then those same cells that were producing antibiotic resistance genes, their daughter cells, most of them will migrate to the steady state where they don't produce the antibiotic resistance genes. And in that way, they will have advantage compared, for example, with different bacterial species, because they will grow much faster. So that's a way in which noise is used as an adaptive mechanism. And actually, this kind of mechanism is called persistence. So these cells are able to, these bacterial species are able to have a state that grows slower, but it survives the antibiotics. And when the antibiotics goes away, then the fraction of the cells that do not produce antibiotic resistant genes will grow faster. So any question about this? No? So noise can be useful. Most of the time systems are evolved to be robust to noise, but some of the times noise can be used as a source of adaptability. We also generally use stochastic simulations to study systems involving gene expression, especially of proteins or genes that have low copy numbers, either of the protein or of the messenger RNA. And when I say low copy numbers, I'm talking about below thousands. So if it is a few hundred copies of either the mRNA or of the protein, then stochasticity has a role there. But if the copy number of the protein is in the order of thousands, then the expression of those proteins is well approximated by continuous systems of differential equations. Another area where stochasticity is relevant is in ionic transport. So biological membranes have a lot of proteins that are involved in the transport of molecules and ions. And although the number of ions in solution is very high, but the number of channels of proteins in the membranes that transport those ions may not be that high. So normally people are interested in studying small areas of the membrane of the cell where you can have, for example, only a few dozens of channels. And those channels actually, although this model is for protein level, but the activity of those ionic channels has these is usually like this. If there are some techniques that allow you to experimentally measure the activity of ionic channels, and they present a pattern of activity like this, because sometimes they are open, sometimes they are closed, and so it's a very stochastic pattern. And if in a given small patch of a membrane you have only a dozen of these ionic channels, then the pattern of transport of that ion is going to be also very present stochastic fluctuations. Nowadays, there is also a new, there are presently being developed every day new methods to analyze single cell biology. So nowadays we can perform transcriptomic experiments that allow you to measure in each individual cell what is the amount of RNA that is expressed for that gene, for a set of genes. So you have data that correspond to a single cell. And if we are studying single cells, then we are going to observe either those extrinsic sources of noise. So cells will be different simply because they, after cell division, one of the daughter cells got more mitochondria than the other, or some of the components have a low copy number. So we are going to see the result of the intrinsic source of node in the expression of some of the genes. So with the advent of single cell technologies, the interpretation of these patterns of stochasticity is becoming more relevant. And so performing stochastic simulations is a way to check if the results that we are seeing in the lab are compatible with what we know about the molecular mechanisms inside the cell. So the rule is that at least one of the variables of the system, one of the dependent variables of the system, has an average copy number that is below the few hundred copies. If you have some of the components like that, then it should be useful to perform stochastic simulations for that system. You don't need that all the variables in the system have low numbers. It is sufficient that some variables of one variable of the system has low number, and the stochasticity in the time evolution of that variable will propagate to the evolution of the other variables that have higher numbers. So if you have a system with a mixed composition, so some variables have a very low copy number and other variables have a very high copy number, and as you will see next, the simulation of stochastic models is more computer intensive, so it consumes more computational resources. Sometimes it is useful to make hybrid models where you do a mix of continuous systems of differential equations simulations with stochastic simulations. So it's like if you model the stochastic evolution of the variables that have low copy number for a small period and use those values, assume that during that stochastic simulation, the variables with a high number are constant, and that should be done for a small period of time. And then you use the time evolution of the stochastic simulation as an input to the continuous simulation of the system of differential equations. And then you update the continuous variables, feed that back to the stochastic differential equation, the stochastic system, and keep updating the system like that. So you perform alternatively, either the stochastic simulation and then the system of differential equation simulation. And you keep updating each other like that. And you have to do that in small time steps. And that is more efficient than performing the stochastic simulation of the complete system. Any question? No?
No.
Okay. What is the difference? Now we are going to focus on how to simulate, how to perform a stochastic simulation. And I expect that next week we are going to try to implement these algorithms in the class, in the practical class. So the model is defined with the same. You can start by defining a system of differential equations. That's a good starting point. Before you do the stochastic simulation, you write down the equivalent system of differential equations, because the system of differential equations will give you the rates of every process that occurs in your model, in your system. The main difference, so you can use similar rate expressions that you used in differential expressions, but the main difference will be in the units of the variables and of the rates. So in ODE systems, in differential equation systems, the variables normally have concentrations of, have units of concentration, so molarity, for example, while in stochastic models, usually we use the number of molecules as the unit. So we don't use concentration. Usually use the number of molecules. And consequently, the rates in the continuous differential equation models are in units of concentration over time, so molar per second, for example. And in stochastic models, instead of working with rates, we work with propensity functions. And the propensity is a probability. And it measures the probability that one event of that reaction will happen in a time unit. That can be a second, a millisecond, what depends on the process. Quicker processes with higher rates will have a higher propensity, and you can enlarge or shorten the time unit that you use, because you shouldn't have very high probabilities of occurrence of an event, because that will give you problems in the simulation. So instead of having rate expressions, we have propensity expressions. And propensity is a probability that goes from 0 to 1, that tells you how probable is that one of those events will occur in a given time unit. So that a change of units and the change of meaning. So a rate tells you the average number of events per unit time, and the propensity tells you how probable it is that one event will occur in that unit time. So you have to, if you have the system of differential equations, and if you have the parameters, you have to convert those parameters in terms of units, convert the concentration into the number of molecules. So you need to know the volume where the reactions are taking place in your system, and multiply the concentration value by the volume, and convert the number of moles into the number of molecules by multiplying by the Avogadro number. So you see that for a large number of molecules in biological systems, if you do that, you get to a gigantic number. And if you have a gigantic number, then you should keep modeling with the system of differential equations. But in cases where after this unit conversion, you get to a number of molecules per cell below a couple of hundreds, then it is useful to perform stochastic simulations. So you need to define what are your variables, what are your dependent variables, and for each dependent variable, you have a propensity function. Okay? You also need to have a stoichiometric vector that will tell you for each process in the system which variables are degraded or produced. And that is equivalent, more or less. You can get that stoichiometric vectors by looking at the differential equations or at the schema of your model. So that's the basic things that you have to know. You have to know which are the dependent variables, which are the processes occurring, and for each process, what is the propensity value, the propensity expression that will tell you the probability that that event will occur in a unit time. Yeah, that propensity function will be a function of the values of the other variables and of parameters that is similar to the rate expressions. So it could be, these propensity functions could be similar to mass action laws. So it will be a parameter, a constant multiplied by the number of molecules of the reactants of that process. So that is similar. And you can also have other types of propensity functions as we used in differential equations. The difference is in the units. But that once we have that propensity function, how do we compute which events will occur at each time? And for that, we have a property that is observed in chemical processes, and in fact, it has been observed in other types of processes. That is, these reactions follow a Poisson process. Okay, so a Poisson distribution is a statistical probability distribution that tells you if a process has a constant rate over time, we can count, for example, during 1 minute, how many events of that process occur. And that number of events per unit time is a Poisson variable. And there's a simple distribution that has one unique parameter that is usually represented by lambda, and that lambda is the average number of events per unit time of that process. And our biological processes, our chemical processes can be considered Poisson processes if the rate of that process is constant over time. But we know that in our systems, the rates of the different processes are affected by the variables of the system. And so what we are going to do is that we are going to assume that we compute the propensity function for the process, and that is going to be valid for a short time interval. And within that time interval, we can compute how many events of that process will occur in a small time step. And actually, for variables that follow a Poisson distribution, we cannot compute also another probability distribution, that is the exponential distribution, that gives you the time between consecutive events of that Poisson process. So whenever you have a Poisson process that gives you the average number of events, or that gives you the random number of events in a time unit, you, in parallel, you have an exponential distribution that has an average that is defined by 1 over lambda, that gives you the distribution, the statistical distribution, or allows you to use a random number generator to get one random time between 2 consecutive events. So sorry, my connection went down. When did you stop listening?
The last thing I remember.
Let me get the slides again. So are you seeing the slides?
Yes.
So I was explaining the exponential distribution that you were listing, right?
Yeah.
Okay, so so the exponential distribution will give you the random distribution of the times between two consecutive events. So if you have a lambda of 1, the average time between two consecutive events will be 1, and actually it will be the most probable the most probable time will be around 0. The times, so most of the times will be below, close to 0 between two consecutive events, and then as we get longer times, those times will be much rarer, less frequently observed between two events. If you increase the lambda, so you get more, a higher number of events per time unit, then on average, the times between two consecutive events will be lower. So we will have a higher probability of times close to 0, and that distribution decays exponentially. On the other hand, if you have a lower average number of events per unit time, then you tend to have longer times between two consecutive events. And so since you have this distribution, you can generate random times between two consecutive events. Another useful property of this propensity, so of this exponential distribution, is that if we have two or more processes with different lambdas, with different average number of events per unit time, you can estimate the joint Poisson process, where if you compute now the probability or the number of either one of the process, so you have two Poisson processes, you have a process A and a process B. Process A follows a Poisson distribution with lambda 1 average events per unit time, and process B has lambda 2 average events per unit time. So lambda, the probability of observing either A or B follows a Poisson distribution with an average number of events per unit time equal to lambda 1 plus lambda 2. And in the time scale, you can also have an exponential distribution describing that processes, giving you that the time between two consecutive events, either A followed by another A, or A followed by a B, or B followed by another A or a B. The distribution of times between two consecutive events will be follow exponential distribution with the parameter lambda 1 plus lambda 2. That means that the average time will be 1 over lambda 1 plus lambda 2. So you can compute these random time intervals between two consecutive events, not just for one process, but for a set of processes. Okay, that's what we are going to use in these basic algorithm to perform stochastic simulations. So that basic algorithm, the first one developed to make these stochastic simulations, is called the Gillespie algorithm. And so you can follow this algorithm in these 7 steps. So first you have to, your system is in a given state, so you know the number of each molecule in your system, the number of each variable, and that is going to be represented by a vector n, that has the number in each position is a different, of the vector is a different variable of the system, and that vector contains the number of each of the variables of the system, of the number of molecules of each of the variable of the system. But then you have propensity functions for all the processes, and you are going to compute a p vector that is going to have the propensity values for every different process that can occur in your system. And that vector is going to be computed with the propensity functions using the values of parameters and the values that are within the vector n. So P is computed as a function of n and also given the parameters of the model. So after you have the vector P computed with the actual state, the present state of the system, that is the vector n, you can sum all the propensity values. And by summing all the propensity values, you get a major P. And having this major propensity value, so this propensity value is the probability that an event, one of the events in the system will occur in the next time unit. So it is like the lambda in the Poisson distribution is the average number of events that you expect to observe per time unit. So using this P, you can use an exponential distribution with this P as the parameter and extract we extract the time to the next event that is a random number from that exponential distribution. Okay, so you compute the time until the next event. So you know that the time will advance until you add the given value of time, and the next time step that you are going to change the state of the system is going to be at time present time plus dt. So and you arrive at a new time point. And at that new time point, you are going to generate another random number, this time a uniform random number between 0 and 1. And that random number will allow you to choose in a kind of a random contest what is, of all the processes that your model has, what is the one that is going to occur at that time point. Okay. So we are going to choose which is the reaction that is going to occur. And once we have shown which reaction is going to occur, then we can update the vector n by summing the stoichiometric vector of that process that was selected. Okay, so we have updated n, so we can go back to step 1 and compute the next time step. So each time step has a different value in the Gillespie algorithm. That different time step is computed from the exponential, is a random number from the exponential distribution. And the parameter of that exponential distribution is the function of the sum of the propensities of all the processes in your model. So how does this random choice of the process occurs? So imagine that you had you were at a time, a given time. You generated the time to the next event using the sum of the propensities, and you get the new time step. That's one thing. That was your original state. So it was a model with 1, 2, 3, 4, 5 variables. So you have 5 different molecules in your system. And here is the vector of the propensities that is arranged in a way that the size of each rectangle is proportional to the propensity. Okay, so the size of the rectangle is proportional to the propensity of, in this case, of process 1 divided by the total propensity, the sum of all propensities. So this is this second process. Given this state of the system, this second process was more probable to occur than the first process, and so on. Okay, so you have you can arrange these propensities in a vector, and you generate a random number, and the position of that random number between 0 and 1 is going to define which process will occur. So in this case, this is the random number, and it will eat this rectangle. So in these from 0 to 1, the frontiers between each rectangle are dictated by these numbers, the propensity divided of each process divided by the total propensity. And in this way, when you generate the random number, you can choose which is the process that occurs. Another way to represent this is like in a wheel of fortune, where each process has a slice of that wheel of fortune with a different area. So the larger the area of the process in the wheel of fortune, the larger the probability that that will be the chosen process to occur. So in this case, the next reaction to occur at this time point is going to be the fourth process. And so you pick up the stoichiometric process, stoichiometric vector for the fourth process. And this process is one that is going to degrade one molecule of this third variable and produce one molecule of the second variable. And so the new state after this reaction 4 occurs. So the first element is not changed. The second element is going to increase in 1 unit. The third element is going to decrease in 1 unit, and all the other elements will going to be constant also. So it is in this way that you update the state of the system. With this new state, you can compute new propensities. So in the next time step, eventually the propensities of the different processes will be different, especially the ones that depend either on the second variable or on the third variable. Those are the ones that are going to change their propensity. And so you can repeat this cycle that you need to generate 2 random numbers, one from the exponential distribution and other to choose from a uniform distribution to choose which process. So there was some question in the chat. Oh.
I think it was just saying what.
Well, what was the last words that I saw? Okay. So Paulo asked if can we assume that the stochastic models are a generalization of the deterministic ODE models? I would say that stochastic models are not a generalization. Stochastic models is like we are really simulating what is happening molecularly. And the differential equation models are, I wouldn't say a generalization, but are an approximation of stochastic models when the number of all the components is sufficiently large. Okay, so actually it is the stochastic models are closer to reality than systems of differential equations. But systems of differential equations give you a very good approximation when the number of molecules in the system are larger. Okay. So have you understood how this Gillespie algorithm goes on? Any question? No? So the problem with the Gillespie algorithm is that so in each time step you have to compute 2 random numbers, and the time step is variable. So you will have the values of the state of the system at not at regular time steps, but at variable time steps. You should be take care in the parameterization, in the choice of the time unit of your model, because if these propensities are the sum of all the propensities is very large, that means that each process is so likely to occur in that time step that probably some events, more than one event will occur in the next time unit. Okay, it is more likely that multiple events will occur simultaneously, and the Gillespie algorithm is exact if the sum of all the propensities is a low number. And by low number, I mean something below one. Okay, because if you have a propensity that is very high, it means that it is very likely that you have multiple events occurring simultaneously. And if for the Gillespie simulation to be exact, you need to model or to decide at each time each individual event occurs. So you cannot have different processes occurring simultaneously. So you can fix that because you can choose in your propensity function, it will have different values depending on the time unit. So if you compute the propensity function for the probability of that event occur in 1 second, that will be probably a very high number. But if you compute it for 1 microsecond, then the propensity will be much lower. So you can model the time unit of your system in order that the sum of all the propensities during all the simulation will be very low. Okay, so that's a way to make sure that the Gillespie simulations are provide exact results, and that means that the propensities must be computed, and therefore, if you choose a much smaller time unit, that means that the times will evolve much slower. If you want to make a simulation during 10 seconds, and if your time unit is microseconds, there will be millions of steps until you made the simulation with 10 seconds. Do you understand that? So that's why it is very computational intensive, because if you want to make sure that you are working with the time unit that ensures that the sum of all the propensities is low enough, that will mean that the time step will evolve very, very slowly in really tiny steps. And if you want to model the system for a larger time interval, that will mean that you have to repeat this cycle millions of times. And that's when it starts to be a slower simulation. So in order to try to make stochastic simulations that are less time consuming computationally, so this new other algorithm was developed. It's called tau leaping, and here, instead of computing the random time to the next event, what you do is you assume that you have a fixed or variable time step, but you define the time step. The time step is not defined by a random number from an exponential distribution. So you choose the time step, and then you compute for all the processes in the model what is the likelihood that they are going to happen in the next time step. So you compute their propensities. And with their propensities, instead of using an exponential an exponential distribution to compute the time to the next event, you generate numbers from Poisson distributions. So for each process, you compute how many events of that process will occur in the next time step. And you can do that for all the processes. Okay, so it starts with computing all the propensity values for all the processes. And so you generate the vector P that is dependent on the vector n for the present time. Then you choose the next time step, and that could be a fixed value, or depending on the value of the propensities, you can, if you have very high number of the propensities are very high, you may want to choose a smaller time step. If your propensities are very low, so there is a low probability of an event happening in the next time unit, then you can choose a longer time step. And then you, for every process, you generate random Poisson numbers for each process, according to the propensity of each process. And that will give you how many times each process will occur in the next time step. Okay, and so you increment the time by the chosen time step, and you increment the state of the system by summing not just one stoichiometric vector, but some for each of the reactions that occurred, and each reaction can occur more than one time. So you multiply the number of times each reaction occurred by each stoichiometric vector, and you sum that result to the previous state vector. And you do that for all the reactions that occurred. So you update the system state, but in the same time step, you can have more than one process occurring, and each process may occur more than one time. So these also simulate the evolution of time of the system stochastically, but it is not as exact as the Gillespie algorithm, because we are computing the propensities. Sorry, a plane is going by. So you are computing the propensities to that tell you how probably, with what probability, so how likely each process will occur in the next time unit, with the vector n at your present time. But during this time step, several processes will occur. And if the number of processes that occur is really high, what happens is that in reality, those processes will not occur simultaneously. So some of them will occur before than others within this time step. So actually, when some of these reactions occurred, their propensity will not be exactly the same, because if some of the process already occurred, they may have changed n, although we are not computing that intermediate change. And each time one of the values of n change, potentially the values of the propensities change. So in this case, we are only updating the n vector after more than one process has occurred, and that may have an impact. That impact depends on how the propensities are changed by the values of n. If they don't change a lot, then it is a good approximation. So it is you want to speed up the simulation, but you have the cost that you are not using the exact propensity values for the time at which the event occurred. So you have the propensity a little bit before the event occurred. Was that clear?
Yes.
Yes. So the tau-leaping is quicker. Gillespie is more exact. Why? Because every time a reaction occurs in the Gillespie algorithm, n is immediately changed, and consequently all the propensities are changed. But to do that, you advance the simulation in random time steps, and those time steps are exactly the times between consecutive events. In the tau-leaping, you speed up the simulation, but you have less accuracy, because you don't update the propensity functions immediately after each event occurs. So you only update the propensity functions after the time step. And in the meantime, during that time step, several processes may have occurred. So you can control the accuracy of the tau leaping by controlling the time step. If you use smaller time steps, you have more accuracy. But by using smaller time steps, you increase the time for computing the simulation. So that's a compromise. And one way to try to adjust for that is using in the tau leaping, using an adjustable time step. So, so as you compute the propensities, if you have the sum of the propensities is very high, it means that if you use a large time step, a lot of processes will occur simultaneously, and so you have more error by using the same propensity functions, although a lot of reactions are occurring. So if you have a large capital P, you should decrease the time step to be more accurate. But if the sum of your propensities is very low, you can speed up the simulation by increasing the time step, because you know that during that longer time step, you will not have so many, you have a low number of reactions occurring or processes occurring. So you control in that way that you are using propensity functions with an accurate, sufficiently accurate value. Okay. Questions?
I would like, do you have a good reference for all this? Because I would like to.
Yeah, of course.
Maybe,
I will translate the slides and
that would also be nice.
In the Brian Ingalls book, there is a chapter talking about I can try to show you. So I'll have to end the presentation. If I go to Moodle. So there is a chapter only about the stochastic simulation. Yeah. Biochemical kinetics. Yeah, here. Here you have it's a small sub-chapter, but talks about stochastic simulations. I can also provide you there are complete books about stochastic simulations, but mainly they try to explore in which situations the stochastic simulations give different results from the continuous deterministic systems that give different results from the similar models that modeled within the continuous deterministic framework. And also some of those stochastic models devote a lot of time trying to mathematically prove or predict properties of the statistical properties of the simulations. So instead of performing exactly all the simulations, you infer the statistical distribution of the states of the system during the simulation. But that is it's more of a theoretical approach. So here,
I think. So I'll read this and see if
this corresponds more or less to what I have presented. Okay.
Okay.
So with these you can follow. But what I was saying is there is a much more extensive world of people working with these stochastic models and trying to extract information from it, from these kind of models. Although this is especially useful, or this is useful for systems that have a smaller number of molecules. So if you are interested in modeling systems that involve proteins that have a low copy number per cell, or the messenger RNA also has a low copy number per cell, then these stochastic systems are relevant. If you are modeling a biological system where you have larger copy numbers, then you are okay using the continuous deterministic approach of systems of differential equations.
Okay.
But of course, sometimes it is useful, and you get information in the stochastic simulations that you don't get in the differential equation systems.
Okay, thank you.
And next week we are going to try to simulate stochastically some of the models that we have talked before, and check if we can observe those differences.
In the TP?
Yeah.
Ah, okay. So the TP would not be for the...
If you have doubts about the report, of course, I can try to help you.
Okay.
But I would like you to, because these, I think it will be much more clearer when you see how exactly this is computed.
Okay, thank you. I think that for me, like I've got a lot of deadlines, so this week I will not be really able to work on it. But will it be possible also to do a meeting outside of the TP if I've got doubts?
About the report, about the paper?
Yeah, exactly.
Yeah, of course.
Okay, then I'll email you.
We just have to find the time for that.
Thank you.
Is it also possible to like send you the paper until the end of this week, and you give like a feedback of adjustments?
So you want me to correct your report twice?
Yes, exactly.
So if I do that for everyone, I have the double of work. So please, if you have doubts, you can ask me.
Okay.
But that would be too much. So I already have some works to correct from other courses that are delayed. So please,
All right.
I will correct when questions you have. I will help for sure. But don't ask me to read the report twice, please. So about the stochastic models, it was sufficiently clear.
Yes.
Maybe not, but I think with the practical examples, it will be more clear. And next week we'll talk about a different kind of modeling so when we started with the systems of differential equations that are by far the most commonly used. Okay, so it's the standard to make simulations of biological systems. Now we went to the stochastic that goes into more molecular detail. So you know exactly at which time the number of molecules changed, and you are seeing changes in the discrete changes in the number of molecules. But of course, you cannot do this for very large systems and for longer simulations, because that is computationally too expensive. But now we are going on next week in the theoretical class. We are going on the other direction. Is that now I want to study larger systems, and I don't have enough detail to build a system of differential equations because I don't have information for all the kinetic parameters or for all the rate expressions. But I want to have an idea of how the system is regulated, and I'm going to focus on regulatory interactions. And that will allow me to simulate the dynamics of the system with a logical network, with a Boolean network in some cases, if I consider that all the variables are either active or inactive. And so we are going to approach next week logical models that are a simplification of the dynamics of a biological system. So this week we went into more detail. Next week we'll go into a more simplified analysis of the dynamics of biological systems. Okay, that's it. Any question?
Teacher, the test will be online or presential?
I haven't had an answer about the room, yeah, because I asked for the room, but nowadays it used to be quite easy to get a room, but I'm not sure if I'm going to be able to get a room out of the period, the defined period for the exams.
Okay, I'm asking because one of our teachers, the exam will be via Moodle, I guess.
Yes.
And that, well, I'm asking about.
Now, eventually, if I do not get a room for the day that we were planning, so it was one day after the end of the first period of exams. So I guess exam starts at 12. So the idea was that we would have our exam in what date?
I think it was scheduled for the 25th.
25th, right? 25th.
Yeah.
Yeah. So I'm not sure if I'm going to if they are letting me book a room on that date, because I sent I made the request, but I had no answer yet. I had to call them again to see if there is a problem. But I'm betting that they are not liking the idea of having a room out of the period, because they want to test the students that go to make before you start doing exams presentially. It seems that you are going to have the possibility to do a rapid test, a rapid COVID test. And so they want to control that more strictly. So I hope next week I have news for you about that.
All right. And what if in the first phase he needs to be online, on the second he also needs to be online? Or can it be present? How is it?
So
I'm sure I won't be able to go to the first phase.
Yeah, if the exam if the first date is going to be online, then I will do all of them online for all of the dates to have an equal type of exam, because the questions are the same. The difference is in the room presentially. Usually, I'm not so worried about the time. Okay, in online, I will control the time you have to do the exam more strictly. That's the main difference, but the questions they don't take too long to answer because they are multiple choice questions. They are not, you don't have to solve a big problem to get to the answer. It's just testing about things that we talked about for example, the network motifs. I can ask you, what does the incoherent feed-forward loop does, and I have several options, and you and you have to check the ones that are correct for the incoherent feedback loop. Things about, for example, the shape of a Hill equation, what does the parameters of a Hill equation mean. So they are questions that shouldn't take you too long to answer, but of course you like to have more time to think and to rethink about the answers. But in the online version, I think last year, sorry, it was the maximum 2 minutes per question. And I would say that the text in the question is not long. So the problem is not reading the question. So it worked okay. The online version last year, I was happy with it, and people had similar grades that they had in previous years. So it worked fine. I would like to do it presentially, but I haven't got an answer back, so I'll try to contact the services again to check if they are going to give me a room or not. If they proposed a room on the 24th of May, is that okay for you?
I have an exam, I think. Let me just. Yeah. Yeah, I have one exam that day. So it would be kind of complicated.
Yeah, because that was the second date more that you chose was the last day.
Yeah, 24 we have your exam as well.
Yeah. Of course. That's a problem, right? Yeah, if I'm not able to have a room on the 25th, probably it's going to be online.
I mean, 2 minutes is not that bad to reply. Like, I've seen worse to not be able to go back. That is really problematic. But 2 minutes and be able to go back, it's not bad.
No, people were able to go back in the last year to check the questions again. Okay, more questions? Okay, so I'll try to check that thing about the exam until next week, and that's it. See you next week then.
Thank you. Bye bye. Have a
Bye.
nice weekend.
Thank you. Bye.
Bye bye.
Bye.